kind of formed an alliance as well now that have announced a opportunity pool program. So it's a cool program for resource development. I encourage you to go take a look at that and contact the O'Brien Centers if you want to get involved with that. Again, if you have positions available, postdocs, faculty, graduate students, whatever, looking for people in your group, please consider using the PKD-RC as a site for advertising of that. Again, visit our site. We have a lot of reagents that change routinely and are updated routinely. Go up and take a look at those. And then contact us if you have any questions regarding PKD, research that's going on in PKD, or resources that you'd love to have us start considering or to build. We want to hear from you guys. So please reach out to us. We're here to help where we can. Today we have a Dr. Bessie from Yale, assistant professor for Yale, who's going to present this roundtable. She will be talking today about DNA JB11, kidney disease, questions answered and questions remaining. So Dr. Bessie, I'm going to turn it over to you.
All right. Thank you so much for inviting me. Let me go ahead and share my screen. All right. Do you see the right form of that?
Everything is good.
All right. Great. So I'm going to talk to you today about DNA JB11 kidney disease. I thought I would go over, sort of synthesize what we know about this disease, because there's been a lot of publications recently. And I've had an interest in this myself since we contributed to what was really a multi-organization institution implication and validation of this as a disease gene. And we recently published some mouse model investigation that I think lends towards us starting to understand the mechanism. So as you all know, ADPKD is the most common monogenic kidney disease. It's the cause of about 5% of end-stage kidney failure. And it's characterized by the gradual accumulation of cysts and expansion of those cysts until they crowd out the renal parenchyma and eventually lead to a decline in GFR, to kidney failure. I have listed here a number of references, which all have genotyped cyst epithelium. And if you look just at the genotype of cyst epithelium, you may think this was a recessive disease because particularly this top reference here by Zhang, published in JASN 2021, they did whole genome sequencing of cyst epithelium, and in 93% of the cysts were able to find what looked like a compound heterozygous genotype, suggesting that at least in a large percentage of the cysts, the cellular recessive mechanism may be at play. We also know from Steve Salmo's recent work that if there was a way to re-express polycystin, potentially those cysts could shrink back in if that was done soon enough, where there hadn't been too much fibrosis around the cyst before that's happened. And I bring that up because today we're going to be talking a little bit about fibrosis around cysts. In order to model ADPKD, many of us use mouse models, and there are a few differences that I wanted to clarify up front. So mouse models, a mouse with a PKD1 heterozygous genotype in the year lifespan or so that it lives in our mouse room doesn't get more than a cyst or two. And we attribute that to the lack of enough time to accumulate somatic mutations. So to get around that, we use conditional alleles, where the flox allele in the presence of Cre can become a null allele, and we can use different Cres to inactivate the different segments or at different times. And that obviously looks a little bit different than what we outlined for the human, in that all the cells in the affected region will have gene inactivation. Of course, there are also hypomorphic models where very mild PKD mutations are subtle enough to be able to exist in the recessive genotype and can be a sort of more recessive model. So as I've alluded to, the disease genes for ADPKD are PKD1 and PKD2, but usually about 8% have no mutation detected, and that's probably some combination of missed mutations, which may be, for example, intronic splice variants or something that we don't catch on exome sequencing or on genome sequencing, don't know how to interpret, or other disease genes. And particularly if you define this ADPKD phenotype either early or inclusive of very mild or liver-specific cases, this fraction that's other becomes much wider. PKD1, PKD2 encode polycystin 1 and polycystin 2, which are complex multiple transmembrane spanning proteins, which need to form a complex traffic to this primary cilium, and that is a membrane-bound organelle that extends from the tubular epithelium into the lumen of the nephron and fuels the flow of urine. So now I want to introduce you to this other phenotype, isolated polycystic liver disease, or ADPLD, where you can see this phenotype has identical histologically, radiographically, and clinically liver cysts. But as you see here, no cysts in the kidney. And turns out that lack of cysts in the kidney is very specific for the lack of PKD1 or PKD2 mutations. And instead, this phenotype has a larger piece of the pie that is genetically unresolved. But through study of this, we've identified several different disease genes, we being a broad we, and a combination of the unsolved piece of that pie and the no mutation detected group of ADPKD, which turns out to be sort of a phenotypic continuum and mechanistic continuum, have led to the implication of many more disease genes, and I've provided the references for lots of nice papers there. So validation of those additional disease genes, I'm going to show you Sec63, for example. This is a 3-week-old mouse that's lost Sec63 in the distal nephron, and you can see already at 3 weeks of age, there's cysts throughout the kidney. And if you take cells out of that kidney and activate Cre in vitro, you can see that there's significantly less polycystin cleaved mature fragment. And so it's, this led to the hypothesis that less polycystin leads to potentially cysts formation. And to validate that, this is Steve Salmo's group with Serin Fidelis, lowered the PKD1 copy number or gave the mouse extra copies of PKD, and less PKD1 led to more severe cysts, whereas if the mouse had 3 extra copies in the form of a bacterial artificial chromosome, that was enough to overcome the concurrent inefficiency and maturation of that protein, enough to avoid cyst formation. So this really showed that the cysts that form in the setting of the loss of at least Sec63 here result from insufficient polycystin function. And this led to the premise that other disease genes, if the homozygous loss of them in vitro led to significant reductions in polycystin, that they could likely be validated as disease genes if found in patient cohorts and other support. You'll see here that the degree to which loss of the disease gene results in polycystin loss doesn't necessarily correlate with the size of cysts. I point out here that Sec61 beta loss results in almost undetectable levels of polycystin, and yet a very healthy person with just tiny little liver cysts. So this often led me to hypothesize that potentially in those same cyst epithelial cells, polycystin is not the only thing that's lost, and potentially other things that are necessary for cyst expansion may be lost as well. So I've been mentioning a bunch of different disease genes. Let me clarify what each of them does. So almost all of the disease genes that I mentioned in that pie chart encode enzymes in the endoplasmic reticulum, or other proteins in the endoplasmic reticulum. ALG8 and ALG9 and GANAB and PRKCSH are all involved in N-glycosylation. These form the N-glycan precursor. That's a sugar that's put on the N or asparagine residue of proteins. And then these enzymes remove glucoses later, before and after the Nexon calreticulin cycle, playing a role in protein folding, preventing protein aggregation, and marking a protein as correctly folded in order for it to traffic. If you zoom in on this location, this is the translocation pore or translocon. You can see the 3 genes I have highlighted in red are implicated in being necessary for polycystin and disease genes, therefore. What I'll note here is that DNA GB11, similarly to Sec63, both have a J domain, which allows them to interact with the ER luminal chaperone known as BIP. I'll also mention that DNA-JB11, unlike most other ER proteins, lacks a ER retention motif, so it's possible that it could traffic outside of the ER as well. I don't have, for the sake of time, I won't go through this case in detail, but this was the first case that I was aware of, thanks to York Pay, who sent us DNA from this family. And I just wanted to point out that this patient had a DNA-JB11 heterozygous truncating mutation. And interestingly, her sister also had liver and kidney cysts. Her mother only had liver cysts, and her uncle only had kidney cysts. Just to highlight the fact that the organ distribution remains a bit of a mystery, probably having other contributing factors, but the same genotype can go either way. So we made a knockout cell line with DNA-JB11 and an epitope tagged, C-terminal epitope tagged cell line. And you can see here, similar to the other genes I've showed, a significant reduction in polycystin C-terminal fragment. But what was unique to this gene, different than any other gene I'd seen, was that the full length didn't stay the same or decrease. In fact, it increased. And the way I interpret that is that if there's less of this and a concurrent increase here, suggests that there's some inefficient cleavage. This is the full length and this is the C-terminal fragment. And that was interesting because it was new and different. And looking from the N-terminal antibody, you can see that here the full length is indeed increased in the null, and the mature N-terminal fragment NTR is almost absent in the null. So this suggests that maybe DNA-JB11 and the ER lumen may be playing some role or somehow helping polycystin cleave. I want to mention here the unfolded protein response because I've mentioned this protein BIP, and I want to show this cartoon that BIP is sort of the main player in handling unfolded proteins in the ER lumen. When there are unfolded proteins, BIP will dissociate from these different unfolded protein response effectors and allow them to aggregate and subsequently produce their downstream consequences, including ATF6 cleavage, XPP1 splicing, and other factors. I partly mentioned that because the Sec63 KSP model, whose kidney I showed you earlier, that in the setting of Sec63 loss, there's activation of the XPP1 branch of the unfolded protein response. And interestingly, Steve Somlo's group with Surin Fidelis looked to see whether that XPP1 activation was helpful or harmful in the setting of the Sec63 model. And so they inactivated both Sec63 and XPP. And it turns out when you lose both, the cysts get worse. And as you see here, the polycystin fails to cleave. So this was sort of the only other time where I've seen evidence of ER protein loss leading to failure of cleavage with PC1. And so that remains an interest to us. In case you think this is just a mouse cell line phenotype, which would be a fair question, we looked back at the original data from the Mayo Clinic cell line in human cells, and there you can see the significantly reduced mature N-terminal fragment in the null cells here. And you see this band here, which is very close to the size of the NTR, but it's actually the full length, as evidenced by the fact that it is endo-H sensitive and moves down to here. So similar to our cell line, you see the full length and the immature N-terminal fragment, but not the mature one. And this full length is increased compared to that seen in the wild type. All right, so now I want to go through the human phenotype because this is really interesting. I've been talking about polycystin-dependent cysts, but this looks different than ADPKD. Granted, I put the information next to these images that have been published from the reference here. And, some of these images are around the time of ESRD, and we do need to be careful of that when looking at imaging because kidneys can shrink after ESRD. But you can see even in those who have declining GFR, but not kidney failure yet, they have normal sized kidneys and the cysts are small. Here's some more examples. This seems to be the only one image published that I've seen where there are enlarged kidneys. These are 19 centimeter kidneys. Otherwise, these kidneys are all basically normal in size. And the cysts, for the most part, are on the small side. Here's another image. And you can see that the GFR, unlike most of those other ER genes, some of which only even cause liver cysts and never have been reported to cause kidney failure, here it seems like there is overall a decline in GFR with age, almost routinely with just a few exceptions. Here's the published available histology of DNAGB11 kidney disease. And here what's remarkable, you can see with the trichome stain with the blue, is fibrosis, how you don't even really see cysts in this field, other than maybe here, but the interstitium is just full of the blue fibrosis. Here there may be, I might call that microcysts or something, but that's surrounded by a lot of fibrosis. And here are the published DNAGB11 pathogenic heterozygous variants. I put in red all of the truncating ones, and so you can see the trend here, that it's not just one mutation, and it's not just missense mutations, it's truncating heterozygous mutations, which makes me think of either a haploinsufficiency mechanism or that somatic mutation, focal recessive of ADPKD. So keep those mechanisms in mind. I want to mention autosomal dominant tubular interstitial kidney disease, because the first publication of DNAGB11 hypothesized that potentially this disease, based on the smaller kidneys and fibrosis, may be an overlap of the pathogenesis of ADPKD and ADTKD. ADTKD, the most common forms are caused by mutations in UMOD or mucin 1. But the specific mutations, so specific missense mutations in UMOD that make it aggregation prone, or a specific frame shift mutation in mucin 1 that leads to an aggregation prone neopeptide. And when those aggregate in the ER, it leads to activation of the unfolded protein response. And that, although the mechanisms may not be clearly fully elucidated here, seems to lead to fibrosis over time. And so I thought it was interesting that here's ADTKD, and then now we have implicated DNAGB11, and there was a publication of Sex61A1. This is the image for it, the third one down on the right, where those are implicated as causes of ADTKD, leading to the hypothesis that, potentially if you either have highly expressed kidney proteins that are aggregation prone genetic mutants, or dysfunction in some of the key ER genes that are necessary either for the highly expressed kidney proteins or proteins homeostasis more in general, that potentially these share a mechanism. I'll also mention that the Sex63XBP double knockout model done by Steve Somlo's group, they actually tried it not with the KSP-CRE, and as I said, it led to worse cysts, but with a different CRE, PKHD1 CRE, it actually didn't lead to much of a cystic phenotype, but instead had these microcysts with extensive fibrosis. And so that's interesting to keep in mind as well, because there the genes were only inactivated in the tubular epithelium, not the interstitium. So to study this more, we made a mouse model. We purchased sperm carrying this allele from UCOM and did the necessary mating to generate a exon 3 flox allele and exon 3 null allele. I'll tell you first about the heterozygous genotype. That's the genotype of the patients, although obviously the patients are getting renal failure in their 70s or 80s, and our mouse we're looking at for a year. Aged over a year, we couldn't appreciate any phenotype whatsoever in the heterozygous mice. We mated heterozygous mice with each other and noticed a lot of dead pups in the cages. But for those first 34 pups that we could count, where we were sure that we had counted the number of live pups on the day of birth, and still that cage maintained the same number at the time we could genotype them. We saw that while there were some null mice that were alive, which is different than many of the other genes I've mentioned, which are all embryonic lethal, this was significantly below the expected Mendelian ratio. We also noticed that these null mice were smaller than their litter mates and would die by weaning age. So because they would die at weaning age, we looked at them at P18. Almost all of the mice that were null and born alive survived to P18. So we were able to look and characterize that indeed they were all lower weight, and their kidney function was abnormal, although to varying extents, as you can see. But the BUN was really only about twice normal, so I don't expect that they're dying from kidney failure. They're probably dying from something else that we couldn't characterize. We looked at hearts and livers and lung, and nothing stood out to me on the histology. We also looked at the embryonic histology and Mendelian ratio, and between E16 and E18, the Mendelian ratio was preserved, so they're dying at some point after that, and the kidney looked fine to us at E16. But when you look in those mice that are born, the kidney does not look fine anymore. It has this interesting atypical cystic phenotype. I would characterize it as actually quite normal looking parenchyma other than the areas of the cyst. And then some of the cysts, most of the cysts have just a simple epithelium, but some of the cysts have what I'm showing here zoomed in. And we saw this in at least 2 or 3 cysts in every null kidney we saw, where there's sort of this multi-layer or blebbing epithelium, which almost was reminiscent to me of a uroepithelium. And in terms of fibrosis, there was none in the unaffected areas, but around the cysts, particularly where there's cysts next to each other, there tended to be fibrosis there. The cysts were derived from both distal and proximal tubule. And looking at the liver, it does seem like there may be a subtle phenotype of increased cellularity around the, excuse me, around the bile duct, but this was not, not even present in all of the portal triad, so it was somewhat subtle to me. The pancreas, we did not appreciate any phenotype. So here's a characterization of all of the null kidneys that we were able to generate. Again, these are hard to come by because of the reduced Mendelian ratio. What we didn't publish, but have data on since, is that we tried to put the PKD1 bacterial artificial chromosome in here with 3 extra copies to see what that did to these cysts. And I think subtly it probably is reducing the cyst burden a little bit, maybe not affecting what looks like potentially some developmental abnormalities. But this kidney and this kidney are litter mates, and certainly this has less cysts. But the Mendelian ratio is still messed up, and they still die at weaning age. So those things I think are not polycystin dosage dependent. All right, around the time that we identified a living null mouse, this paper was published in KI, showing that there was a human fetus, excuse me, with enlarged polycystic kidneys. This was born to an Afghani family that had recurrent pregnancy losses, as well as some healthy children. And so this pregnancy was watched closely and unfortunately had to be terminated due to anhydramnios because basically this kidney is not functioning. Autopsy showed that there's abnormal liver where there's periportal fibrosis and sort of extra portal areas, and fibrosis throughout the pancreas as well. They also proposed that potentially cilia appeared longer in the DNAJB11 recessive human fetus. So we wanted to go on and make a conditional mouse model with the purpose of not wanting to see only the minority of null mice that live, but also being able to look at these mice a little bit older in age, more akin to the human phenotype. So we decided to use the KSP-Cre in order to parallel the previous Sec63 experiment. And much to our surprise, DNAJB11 flox KSP-Cre had no phenotype whatsoever. This is really puzzling to us because the KSP-Cre is active in utero around E10 or so, and it may take a few days for the protein to go away, but still it's not that much later than the null mouse that had the cystic phenotype. And so that led to lots of other hypotheses about what other cell types are necessary and so on. But we decided to, oh, here, I'll show you. I'll show you the phenotype of kidneys aged beyond a year. And essentially, I don't think there really is any phenotype. To me, we never see good-looking kidneys after a year. The only thing that I'm showing here is the only abnormal regions of these kidneys where we see some perivascular lymphoid tissue. But I see that in wild-type mice as well, and I was not able to find any significant increase in these mice compared to the wild type. So we next wanted to put this with a PKD1 heterozygous genotype, which had worsened cysts in the Sec63 model. I'll remind you that the PKD1 heterozygous genotype itself, here there's no CRE, so this is just a PKD1 het, has no cysts. But when we put that with DNAJB11 flocks, so two things that don't have cysts in themselves put together, they do have this robust cystic phenotype. And so that's a model that we've studied quite a bit now. Yeah. In terms of this cystic model, we wanted to look over time. So at 3 weeks, you can see that there already are a good number of cysts. The mouse and the kidney are obviously still getting bigger between 3 weeks and 9 weeks. And so all of that expands. Going to 25 weeks, and here I'll give you the cystic index data. The cystic index no longer expands. If anything, it almost shrinks. And I was struck on this 25-week kidney by these areas that seemed distorted, and with serious red stain are almost what I was tempted to call like islands of fibrosis, almost making me wonder whether cysts had involuted, although there's no staining of cyst epithelium cells that I could appreciate within the areas. Here I'm showing you some of that serious red staining. So here's the 9-week kidney, and here's the 25-week kidney. It seemed like there was significant increase in the amount of fibrosis between here and here. And as you see, no expansion of the cysts. So that may be, similar to the human phenotype. We also wanted to get a sense, though, whether the little bit of a fibrosis that we do see around the cysts in the 9-week kidney, or it's a lot around the cysts. It's just not much in the unaffected areas, was similar to a PKD1 model, or was any different, whether DNAJB11 made it any different. So I got these kidneys from Kerr Dong and Steve Salmo's lab, where it used a WS25 PKD2 model, which has stochastic inactivation, leading to focal cysts. And with these focal cysts, again, I think somewhat similarly to the DNAJB11, there's fibrosis around the cysts, but not in the unaffected parenchyma. So I could not say that for sure DNAJB11 was causing any more fibrosis at this time point, but I didn't have these mice aged any further to be able to draw the comparison here. And I was pretty impressed with the fibrosis in our 25-week mice. So what about the unfolded protein response? I mentioned that as a proposed hypothesis. I think I don't have time to go through all this in vitro data, but we looked at several markers here. I'll show you the in vivo data for the sake of time. We looked at the protein expression of BIP, which should go up in response to activation of any of the 3 pathways. And as you can see here in both the germline model with the null cystic kidney or the conditional model, there's no change in BIP expression. Looking by qPCR, also for BIP, CHOP, or XBP1, there's no difference in our conditional model, the purple being our cystic KSP model mouse. Just to make sure that our assay was sensitive enough to be able to pick this up, we did two different controls here. The S is the Sec63 KSP, and so you can see with that genotype, there definitely is a significant increase. And with the tunicamycin treatment, there's a significant increase. So I think our qPCR was sensitive enough to detect UPR if it was there, but it's just not there. We also looked at uromodulin and mucin and put that in our supplemental data. Those seem to mature just fine in the cyst epithelium that have lost DNAJB11. And this paper came out just recently in KI, where they looked at the uromodulin levels in human urine. 80 TKD UMOD patients are known to have low urine uromodulin because it doesn't mature appropriately to get to the cell surface and get secreted into the urine. And you can see here, indeed, the 80 TKD UMOD patients have very low uromodulin. These normalized to GFR. But the DNA-JB11 patients did not at all have low. If anything, they were a little bit higher than the ADPKD. They used a knockdown analysis in cell lines and showed no difference in uromodulin maturation as well. They did detect an increase in aquaporin 2 and particularly glycosylated aquaporin 2. So in summary, I have talked to you about the effect of DNA-JB11 loss on polycystin maturation, mentioned what looks like a cleavage defect. Talked to you about the extra renal effects, where the DNA-JB11 null mice seem to have null mice and human recessive disease seems to have developmental abnormality in kidney, liver, and pancreas, and early lethality. I think it's important to know that DNA-JB11 null genotype is mild enough that human fetuses will potentially live to the age at which screening would be done and detect PKD, unlike ADPKD. And the renal effects, DNA-JB11 null genotype, I showed you the atypical PKD. We've talked about the human phenotype of the heterozygous and our mouse models where there's no detectable phenotype unless you adjust that polycystin dosage enough to get cysts. We also talked about how DNA-JB11 loss does not seem to affect the unfolded protein response, nor uromodulin maturation. So I tried to put together a little cartoon here because we certainly don't know the mechanism yet, but I think we've learned some important components of it. So here, wild type cells are green, heterozygous cells are yellow, and null cells are red. And I think the heterozygous genotype puts someone at risk for the somatic mutations that we have in our cells every day to happen to hit DNA-JB11 in this case, resulting in the null genotype. And I think extrapolating from what we know with ADPKD cyst epithelium, it's reasonable to think that a DNA-JB11 null cell can proliferate into a cyst due to insufficient mature PC1, and that results in ADPKD-like cysts. But the phenotype isn't exactly like ADPKD, so I feel like there's something else that we need to explain still to explain the fibrosis or restricted cyst growth. And so the question is, what is that? I think what was proposed initially is that it could be haploinsufficiency over time. And I think that very much could still be it, but maybe not via uromodulin or unfolded protein response. The fact that our mouse model didn't have activation of UPR or uromodulin, and in fact didn't even have a phenotype, to me suggests it makes it even harder to swallow that haploinsufficiency is going to lead to a phenotype in itself. It seems like the cysts part was really important, and if I think losing both copies of it can at least try to hurry up what does take years in humans. But perhaps this could suggest that maybe just haploinsufficiency in the tubular epithelium isn't enough, but maybe in other cell types could still be a mechanism. Alternatively, and perhaps supported by the fact that our DNA-JB11 conditional mouse at 9 weeks was no different than the PKD2 one, maybe it's the haploinsufficiency of the cells around the cyst, and that DNA-JB11 genotype responds differently to those cysts. Maybe it doesn't let them expand, or maybe it has a more vigorous inflammatory response and affects the cyst growth that way, or the ability of them to expand. Or alternatively, and I think this would be an exciting hypothesis because it could really teach us a lot about fibrosis and kidney disease in general, is if whatever signal cysts send out to that brings in the inflammatory response that we know comes, whether there's something about a cell that has DNAJB11 loss and insufficient polycystin that might make that cyst epithelium unique in some way, that either in the setting of those heterozygous cells that are nearby, or even if they were wild type cells, whether that could lead to this, whatever's different about this disease. And with that, so we are trying to work on some mouse models, potentially inactivating both DNAJB11 and polycystin to see how either the heterozygous genotype or concurrent loss can affect the cyst progression. But that's in progress, so I don't have much to share about it yet. With that, I will thank my great team of Zhigui Li and Zhe as well as my current and former students, and Shonik Gashua, who did most of the mouse work that I showed today. Thank you to my collaborators, past and current funding, and our ongoing PLD study. Thank you, and I'll take any questions.
Thank you, Dr. Vinci. It is now open for discussion. I guess I can, if you have a question, please unmute yourself and ask. Way it stimulates, I think, conversation. I'll start off with a question. You know, when I think of shrinking kidneys and fibrosis, and small cysts or something, I think of nephronophthisis.
Mm-hmm.
Is there
Which I
any
should have, I can't believe I
connection?
didn't say that word. Yeah, okay.
Is there any connection between your gene and a nephronophthisis genes that are out there that Hildebrand has identified?
So I think I probably should say I don't know, but we did look at NPHP3 in the setting of DNAJB11 loss. I guess here, it doesn't seem to be affected.
Okay.
And I know that that's not the only nephronophthisis gene, so potentially others could be affected. I think certainly anything that would be a membrane protein or a cilia protein may need to, particularly cilia membrane protein, may need to go through the ER and therefore could be affected by DNAJB11. And so I think that's entirely possible. The IVOMARK2 syndrome, the recessive disease, that renal hepatopancreatic dysplasia really is, classically caused by NEC8 or NPHP3. So that really does draw the comparison between this and nephronophthisis genes. So I think that's an avenue that we need to look into more.
Okay. Other questions? Well, I'll ask another one with nobody else is going to. I mean, you showed this genetic interaction between alleles with polycystin 1. Any evidence that polycystin 2 mutants in this background, heterozygous or otherwise, would alter the phenotypes? Or is this specific to polycystin 1?
Extrapolating from what we know from other disease genes, including going from the Steve Somlow's Nature Genetics paper that looked at Sec63 and PRKCSH. There they looked with PKD2, and I think that did not make a difference. It all seems to go via polycystin 1 message. On the same slide that I had showed before, we also looked at.
I saw there was no processing.
Yeah, there didn't seem to be any effect there either. So I think it's via polycystin 1.
Okay. Fair enough.
But we haven't looked at the genetic interaction, so I can't say for sure.
All right, questions? White group.
Come on, guys.
Brad, can I ask a quick?
Oh, absolutely.
Okay. Hi, Whitney. Great talk.
Hi. Thanks.
Quick question about why they don't survive past weaning and whether they just not nurse as much, or do you have any idea? Or could it be some malformation in the gut?
I would have told you if I had an idea. I don't know. I do feel like they fail almost immediately after weaning. So it could be something to do with like their ability to nourish themselves, or they do better when they are with their mother, and as soon as that's gone.
I was just wondering of other ciliary phenotypes like olfaction or something like that, that
Yeah, they are little and funny looking. But I don't know I don't know
Okay.
well enough how to assess those other
is just,
characteristics. I'm open to suggestions.
and no left-right axis issues.
Yeah,
No.
they're fine.
I was gonna ask, the null mutants that don't survive, right? What is that, what's causing their lethality in an early developmental context?
I don't. And we found some null embryos, but I couldn't one of them looked completely messed up. Like I couldn't even recognize the structures, and others I saw looked I couldn't identify I couldn't appreciate anything abnormal.
Because by, going with this small group that is surviving, you're having a survival bias to some of the stuff you're looking at, right?
Exactly. That's why
But
we
it may
wanted to
hide
sort of-
some of the real roles
Right.
that this gene's playing would
Yeah.
be.
And potentially I should have, we should have sent out some of these, or still could send out some of these slides to someone who knows more than I do in terms of that histology, but
Yeah.
that's what we know at this point.
Harry mentioned that maybe gut issues. I'm just wondering, like, what kind of gut issues, Terry, you were thinking about, which might cause the death or be associated with death.
You're muted, Terry.
I was just thinking like the villi and I mean, that, I think it's been reported in other mouse models of other kinds of things. When you look at the formation of the gut, the villi are just not, not well formed.
I see.
I mean, I don't know.
A lot of times if it's an olfaction problem, they don't nurse well either.
Yeah, that's what I was also thinking, just,
Oh, because they cannot smell.
yeah.
But what was the Cree you used? Or they were germline mutants die at 3 weeks?
Germline mutants.
Mm.
There was
Compound
also,
head process.
So it's definitely expressed on the cilia, right?
no, it's not expressed in the cell. It's in the ER lumen. It lacks the ER retention motif, so in theory it could make its way out and not have vesicles bring it back to the ER, but yeah, we only see it in the ER.
I mean, I guess one way to think about this is a general process of maturation of proteins that have to go through the ER. So there's a chance you are really mucking up a lot of different things and not just. So have you looked at, I don't know, CFTR, for example? Is the maturation process of that protein messed up? Or
Yeah, that'd be an interesting one to look at because it's also big like PC1.
yeah, and there's clear
Yeah.
steps in the glycosylation steps that
Yep.
are for its function.
Hi, Chris Guerrero here.
Hi.
Hi, I mean, just following up on that comment, the fact that you don't have an overt UPR would
Mhm.
suggest that you're not really mucking up with a lot of proteins.
Okay.
It is important to keep in mind that, well, sometimes satisfying to think of the J proteins as only acting with their HSP70s as co-chaperones. They do have roles by themselves in protein folding, and they form complex homomeric and heteromeric interactions with other J proteins. So there could just be a set of clients like PC1 that are highly dependent on JB11 by itself or with other J domain containing proteins that may be involved.
Yeah, thank you.
That's input.
Any suggestions on how to work that up? Or
I'll think about
outline
it.
that group of protein?
I will think about it a little bit. There was a great protein or great paper from I forget whose lab it is. I'll send it to you once I find it. Looking at some of these complex interactions between J proteins and clients, that might be interesting to think about.
Yeah.
I assume there's no
have a-
specific motif they bind to, right? That
Sometimes there's fairly specific motifs the J proteins themselves use, but in terms of the clients, there's no specificity.
thanks.
But given that polysystem 1 is such a beast to fold, it's going to make it more sensitive anyway.
Right.
Yeah, Whitney, thank you so much for the great talk.
Thanks.
Yeah, I have a quick question. Looking at your histology of your mouse model, the fibrosis occur in those areas where you have cysts. Yeah, this one. Yeah, perfect. Whereas the blue one is normal, right? On the brown is fibrosis.
The red is fibrosis, yeah.
Red is fibrosis,
brown.
yeah.
Yeah.
So do you think the fibrosis occur because of lack of mature PC1 or it is because of cyst formation?
So usually with PKD1, advanced stage or when there's more cysts, you get some fibrosis.
Right.
We know that. I think that certainly in the human phenotype, we know something is different. And I'm wondering whether maybe our 25-week mouse model here is recapitulating that. In this mouse model, the fibrosis is very much either right around the cysts or in these areas which I wanted to call fibrosis islands. They're just areas of fibrosis where nothing recognizable in terms of cell types. And I'm no expert on inflammatory cells, so I didn't look at the different cell types there. But there's no, LTL or DBA or any other tubular marker staining within these areas. But they all seem to sort of have this central little something that made me wonder if they were a cyst that just involuted. So my sense was that it was sort of an overzealous, if I had to put something on this particular mouse model, it seemed like an overzealous response to cysts.
Okay.
It was focal.
Okay.
Yeah. Another thing which is striking is that the blue cell, what type of cell could that be? I think is proximal tubule maybe.
Blue, you mean the cyst epithelium here? Or?
No, the blue color, the non-cystic cell tubule on the right-hand side, the right panel.
Right here? What?
On the very
I think you stained the whole kidney section with blue, right?
Yeah, the serious red shows up as red, which is collagen basically, and then the counter stain makes the rest of the kidney blue.
Yeah.
Okay. No,
So the blue isn't anything
is
specific.
Tubule. What kind of origin is this?
The tubules in this particular image?
Yeah, on the right of it, right panel.
I
below,
don't
very
have
small.
concurrent DBA or LTL to tell me which.
Okay.
Yeah.
Paul?
Because I mean, but the reason I'm say that because based on biochemistry, your mouse model should have less cleavage, right? In those DBA, DNA JB11 negative cell, the cleavage is diminished.
Right.
So mimicking really a cleavage mutant, similar like the T to V mouse model, as we previously showed, the proximal tubule is completely intact. And looking like your mouse, if that's a blue thing, is a proximal tubule.
Actually, sorry, you know what? The cysts are distal, because in this KSP model, we only inactivate, it's from the UMOD positive segment.
Oh, yeah.
So sorry, I slipped my mind because the null is both types, but the KSP model here, this is distal.
All right.
So is that, you said that you do get distal cysts with your?
Yes, our PV model, which looked similar like to your histology, is only in the distal and collecting that, and not in the proximal tubule. But your explanation is also clear because you use the KSP cree, so you don't know what happened in the proximal tubule, the requirement of it.
Right. Thanks.
Thank you.
Think of that model too.
Yeah. Now, Paul.
Thank you. Really marvelous work. Followed it for several years. I think you're unearthing lots of interesting things to pursue.
Thank you.
Just going back to this DNA JB, what the clients are, I wonder if you've considered proximity labeling to identify at least what could be the initiating factors. There are many others that are downstream that, for example, regulate EGF signaling pathways and such, but at least you could get at the most proximal steps. I wonder if you have marvelous cell lines to begin to ferret that out.
Yeah, but I'd have to look into that more. In what model or system would you do that, the proximity labeling?
Well, I think that's the question for you to figure out. I'm not really sure which one would be best, but I think a model where you have all of the components as you've outlined in your talk and publications would be the right place to start. A place where you really do have altered PC1 abundance or splicing as proteolytic processing, as you showed, and then use that model to ferret out what the other clients are, and then perhaps add them back or in the model to
Right.
see, you know, what they do.
Yeah, no, that's an interesting thought. We have thought a little bit of, you know, just in terms of looking at PC1 DNA-JB11 interaction or other interactors of DNA-JB11. The challenge that I've, at least in my reading, that's made me not jump into it quite yet is just that I think a lot of the interactions in the ER are transient and need, forgetting the term, but basically to do a co-IP with a ER chaperone, you may need to sort of chemically fix those interactions in a way that could lead to some false positive or lack of
The other
specificity.
possibility would to do something with a BRA, which I think is what Paul was referring to, one of these promiscuous ligases that will label things transiently that they interact with, and then you just pull them out and do mass spec. So
Exactly.
You said, or-
BRA, yeah, BIRA, capital A. There's a lot of different forms of those to do proximity labeling to look at interactions between proteins.
I need to learn some new techniques. Thank you.
All right, any other questions? We're right at the hour.
There are
So,
some questions on the chat.
if you would prefer to, I would prefer people get, because this stimulates the conversation. I'm trying to get people to speak up. 2 questions in the chat. Did you ever, Steve Purnell asked, did you ever check blood glucose levels of surviving pups?
I did not, no.
That could
That
be
might have
another-
been an interesting thing just in terms of survival.
Yeah,
Wonder-
and they could go hypoglycemic and be out of there in a heartbeat, but yeah.
Right. I don't know if Steve has any particular reason why he would expect that to be abnormal, or just thinking that it's something that should be generally checked.
All right, and then the other question.
Okay, thank you, Stephen.
Pam Tran, was, do you know if DNA-JB11 plays a role in skeletal development that could cause the small phenotype?
I do not know, but I will say that this small mice looked not just small, but a little bit sluggish and ill. So I would tend to think that it wasn't just that they were small from a skeletal perspective, but don't know for sure. Interesting question.
Steve says his mic isn't working, so he'll email you.
Okay, yeah, I saw it.
Okay. All right, we're right at the hour. Dr. Besti, I want to thank you for a wonderful talk and for taking the time to meet with us. Keep doing the good work. You know, normally I end these by asking if there's something the consortium could do to help with your research. We've got about 3 minutes left. If there's something you want to add that, you know, that we should consider as a consortium, we're here to hear it. So
I wasn't expecting the question. I'm not sure.
that's fine.
Nothing immediately comes to mind. Although I just for some controls and other things, we actually just ordered some cell lines and antibodies from you guys. So
Excellent.
I
Oh,
totally
so
appreciate the PKD RRC and we'll support it.
And you know, if something should come up that would help with you know your projects downstream from this, please don't hesitate to reach out to us, Terry, myself, Steve, Paul. Any of us are here to help advance your research. So keep up the good work. Again, thank you for spending the last hour with us, and appreciate the active discussion from the group. So thank you, everybody.